Hi, this is Dr. Barron. I want to explain to you what exactly you will be accomplishing today. Just as a review of this week, you have been doing the Casey, which is wonderful work, but now we have to transition to making sure we fully understand the structure of DNA and how that structure of DNA results in the production of proteins. So DNA to proteins is the big idea that we've been focusing on. And we're going to be using that understanding to do more biotechnology and particularly genetic engineering. So what I'm going to ask everyone in the class who got below a 3.5 on the test, your substitute should have passed back the tests by now with the scores on them. What I would like to do is I'd like to review with you exactly how you are going to raise your grade on it so that everyone in the class can get above a 3.5 on the test. So the first thing you're going to do is click on where it says Competency 5 DNA, if you haven't done that already, to access the video. And then what you're going to do is you're going to click on what it says right here, DNA to protein test corrections. When you click and open up that document, what I would like you to do is I'd like you to leave it open in the web browser. Unfortunately, if you download it, then you'll lose the ability to click on links. The links are located on the lower part of the assignment, and you need these links to help do part of your test corrections. There will be a hard copy provided to you, and you'll see that there's 2 steps. So your substitute will provide you this handout, and this is where you'll do all your writing is on the paper copy. So it says, step 1, read examples of essays for each paragraph and write down and fill out the improvement plan. Note, you must do this to be allowed to retake the original test. And like I said, make sure you view this in the PDF in a web browser. This will allow you to click on the links for videos. So the very first part is this part. What you'll first see is that for each paragraph, I have taken sample student work and copied and pasted, well actually I hand wrote, or I typed it in, for no mark score, 2.5 to 3 score, and a 3.5 to 4 score. And in the first paragraph, you are asked to answer the questions, where is DNA found in the cell and how is DNA organized? And you are supposed to use these words right here. So what your task is to do is to read all 3 of these, and from all 3 of these, you're supposed to gain a sense of understanding about what you need to add on to your essay to get it up to a 4, and particularly what's the difference between your score and 4. You may be saying the same thing for both of these answers, but in some cases they may be different depending on what you wrote for a particular paragraph. So what I'd like to do is I'd like to show one scholar's paper, and this is Britzy from our class. And what you can see is that overall she got a 2.5. Her first paragraph she got a 3, which meant she mostly understood everything. You'll see that I actually circled the word chromosome. So anywhere on your paper that you see a circle means that you didn't fully define what that word means, or you didn't accurately define what that word means. Some people said there are always chromosomes in a cell, and that's not necessarily true. A chromosome is a single-stranded molecule that only gets that shape when a cell goes through division, when it decides to divide. We refer to them as chromosomes as a single molecule of DNA, which is just a really long strand, and we have 46 of those in normal cells in humans, and we have 23 in our sex cells. So in any case, if you didn't accurately explain what a chromosome is, or you just didn't explain it, and same with the other words on the test, you'll see the other things I circled for her, then the more circles essentially, the lower your score, because you're not fully explaining how those words are used to answer the question. And the first question was, where is DNA found in the cell and how is the DNA organized? So when you look at the no-mark scholar, the no-mark scholar wrote, DNA is found in the nucleus. DNA is organized with prokaryote and eukaryote chromosomes. Now, that doesn't make any sense. There's no difference between prokaryote and eukaryote chromosomes necessarily. Maybe the type of organism determines how the shape of the chromosome is, or how long that strand is, but that doesn't make much sense. Strands of DNA is what makes up a cell. No, cells aren't made of DNA. Cells are made of lots of different types of molecules that have all been made or are made up by proteins. And a genome, so see how this is not accurate at all. That's where the problem is, and that's why it's a no mark. If you look at, so this would be scholars who had like a 1 on If you look at the second paragraph, a cell is a building block of life. Okay, good. That's correct. It sort of defines what a cell is. DNA is found in the cell's nucleus. Well, DNA is also found in cells' cytoplasm as well for prokaryotes, so that's not totally accurate. A cell with a nucleus is called a eukaryote. Without a nucleus is called a prokaryote. That's correct. Both cells still have DNA and are tightly coiled in a thread-like structure called a chromosome. Yeah, but that chromosome doesn't exist at all times. It's only during cell division. It's just a single strand of a molecule is a chromosome, but it forms that shape only during division. The complete set of DNA, including the genes in an organism, is called a genome. That's correct. So this scholar is actually closer to the 3 than the 2.5. But you can see the problem I would have circled is chromosome, and not the most comprehensive understanding of all of these things. If you read the 3.5 to a 4 paragraph, a cell is the most basic unit of life. Cells are what make up organisms. There's a more thorough definition of a cell. See how the author stops and then defines a cell and says how it relates to organisms, living things. There are 2 types of cells, prokaryotic and eukaryote. And by the way, this scholar over here does not necessarily have a 4. Prokaryotic cells have no nucleus where the DNA is found in the cytoplasm. Eukaryotic cells have a nucleus, and that is where the DNA is stored. DNA is organized into chromosomes, tightly packed, and wound up DNA. Humans have 46 chromosomes, and they separate during cell division. So I see he understands that these chromosomes are involved with the cell division, but that was why he wasn't a 4 actually on this first one. All the DNA in an organism's body is known as a genome. So you can see the difference between them. So now what you need to do is you need to write down specifically what the difference is between yours and a 4, and what do you need to add on to make it a 4. Now the next one, going over this as well, you see me go through I can go through the same thing. You'll see inaccuracies and not complete explanations in the NM category. In the 2.5 to 3, you'll see that some things are not fully explained. Like we have to talk about use all of these words to explain the basic structure of DNA. And you'll see this scholar, this means that there's 2 strands that rotate around each other. That explains the double helix. But just saying 3' to 5' and 5' to 3' doesn't define anti-parallel. You'll see instead, this definition makes more sense for anti-parallel, meaning they run in opposite directions, the 2 strands, and the molecules are upside down from each other. So you'll see those differences between each column, and for each paragraph you have to write down your improvement plan about what the difference is between your score and a 4. You've been provided a lot of detailed support on this, and you definitely want to work in a group to better support what plan. And I will be grading this plan, these answers for each paragraph, and you have to have it complete before you actually take the retake. And then also the thing to be aware of is that you have to keep that old test, because you're going to have to staple the old test to this packet and then your new test that you take. Once you finish that, then you get down to this step, and you'll be given images and links to videos on the left, and then sort of a fill-in-the-blank affair on the right in terms of being able to explain the image that you see on the left. The answers for this will be hanging on the whiteboard. If you scroll down a bit further, you'll see that there's some that aren't fill-in-the-blank, and you have to actually explain what's in the image. And then others are fill-in-the-blank, where you have to finish describing everything. And both the videos and the images are there to support you. Once you've done all of this, then you're ready for a retake. And we'll do retakes starting on Friday. If you have any other questions, please let me know. And scholars who do not have below a 3.5, I want you working on your acceleration plan, clearing no marks, or I would like you to work on your project. If you have any other questions, let me know.